Antibiotics brought about a revolution in medicine. Before antibiotics, people would regularly die of horrific bacterial infections. And these could be, you could get infected from something as simple as a cut on your finger. Because of the success of medicine driven by antibiotics, they've been heavily used and actually overused. The problem is that bacteria are evolving living organisms, and in response to the use of antibiotics, they have evolved new forms that can resist the impacts of these drugs. As a result, the effectiveness of antibiotics has plummeted, and now we are faced with many bacteria that we are unable to treat with the typical antibiotics. Part of the solution has been to use antibiotics less. And indeed, Chile has been a leader in Latin America in making sure that antibiotics are only prescribed and used where they're needed. But this has proved insufficient to prevent the rise of antibiotic resistance. And now, people are dying. Indeed, more people are dying from antibiotic-resistant infections than from HIV or malaria. We urgently need new solutions. And what I'm going to argue today, that one of the key routes out of the antibiotic resistance crisis is to think more about bacteria and how bacteria live, and indeed how bacteria affect each other. But before we get there, what do you think about when I say bacteria? Maybe you think about something like this. This is a picture of a bacteria, or bacterium. Now, they're very small. They're about 1 micron long. What does that mean? It means you can lay about 1,000 bacteria end to end on the end of a pin. So they're tiny. Bacteria are also relatively simple. Their cells are not as complex as the cells in your body, for example. But despite being small, and despite being relatively simple, bacteria clearly have huge impacts. But this is not how bacteria really look most of the time. To understand how bacteria really live, we have to think more about something like this. So where they occur in the environment, but also on and inside your body, particularly inside the human gut. They live in dense communities with many different strains and species. And importantly, these species can affect one another. Moreover, while some bacteria do cause deadly diseases, many of them are harmless, and in fact, many of them are also beneficial, and they protect us against the bacteria that cause disease. So what I'm going to argue today is that we need to understand better how these good bacteria function, and indeed harness them in ways that we can treat disease without the use of antibiotics. So the idea is using bacteria against bacteria. Bugs, not drugs. So where are we going to look in the lives of bacteria to try and find new ways to treat disease? Well, bacteria affect each other in many ways. Sometimes they work together as a team, but a lot of the time they're working against each other, they're competing. And so the kind of ecological interactions we're interested in is something like this. So this is a classic example of competition, seen very clearly between two animals. So do bacteria do this kind of competition? Well, they do something very similar. And actually, it turns out that bacteria are amongst the most aggressive organisms we know when it comes to fighting with other bacteria. So what does a bacteria fight look like? Well, here's a computer simulation from my laboratory of bacteria who are at war, who are fighting. And so in this movie, the green bacteria are using a tiny spear that they fire out of their cells into other cells. This spear carries a toxin. Now, if it's the same type of bacteria, they'll have the antitoxin and they survive. But if this spear hits a different type of bacteria, it can rapidly kill it, and it helps it to compete. But this is just a computer program version. We can also see what this looks like in the lab. So here's another movie, now with real bacteria, showing the same thing. So you can focus on the top row where the colors are. That shows you most of what's going on. The green bacteria here, again, carries one of these spears, these poison spears, and it's firing it into the pink cells. And you can see it's having a very decisive effect on who wins in this competition. So these bacterial weapons, as they are known, are extremely important in deciding whether or not a given strain of bacteria will be present in a given environment. I'd like to give you just one more example of bacteria undergoing competition. And in this, they're fighting as big groups, as big teams. So in the movie I'm going to show you, there's now millions of cells involved. On one side, you have a bacteria that makes a toxin, its own type of antibiotic that can target the other side. And on the other side, that also has a toxin that can target the other strain. But they are immune to their own toxins. Importantly, though, this is not just about them blindly releasing toxins into the environment. The bacteria do more than that. They are able to detect the incoming attack from the other strain and respond in kind. So they reciprocate. They detect and they fight back. So bacteria can display quite sophisticated behaviors when it comes to conflict between different strains. So I'm going to run the movie. Importantly, we've genetically engineered these cells so they go green when they're making their toxins. So this video is sped up. The whole video is about 20 hours. But as you can see, they get green closest to each other. And that's because they're detecting each other, they're responding, they're getting more angry, if you like, and they're fighting more and more with each other. And as the movie goes, you see they get brighter and brighter because they're getting more and more kind of pissed off, if you like, at each other in this competition. Okay, so bacteria fight, and they fight very aggressively. This is not the only thing they do, and it's not the only way they do it. And so one thing we did recently was develop an online computer game in order to illustrate the wonders and mysteries of bacterial competition. This is aimed primarily at children, so if you have children, you might enjoy it. But we also find a lot of people enjoy it who don't have children. But it's a very quick way online to get into the idea of how bacteria compete, and indeed the importance of this potentially for understanding our health. Okay. So that's available to play online via an Oxford website. But, okay, so why is this, so this hopefully is interesting, but how is this useful? Well, it's useful because, as I said, we carry many of these bacteria inside us. And these bacteria that we carry are extremely important for our health, we now realize. The challenge, though, is we don't just carry one or two of these strains, we carry many. In fact, we can carry hundreds, if not thousands, of species. And as I say, it's particularly important in the gut, where they reach very high densities, and they've been shown to be important for all aspects of our health. So how do we begin to understand these micro-worlds with so many different species? We're thinking more like a rainforest now than anything else, many different strains and species interacting in an ecological system. How do we crack that? Well, that's proved challenging, and that's the challenge of understanding the human microbiome. There's so many moving parts, it can be difficult to dissect it and understand why it's good, why it's helping us. So how do we approach this? Well, in my laboratory, we approach this using a combination of mathematics and computation, and then simple experiments to try and understand the causes of the benefits that these bacteria give us. And in particular, I'm going to focus today on this key benefit, which I've been discussing throughout my talk, which is the ability to prevent pathogens from growing inside the gut and causing disease. So, how do we go about this? As I say, we go about this in various ways, but particularly I like simple experiments because you can learn a lot from a nice experiment. So, just to run through then what we do, we focus on, I'm going to just use the word pathogen in this talk, but behind the scenes, we've studied three different important species of bacteria. So E. coli, Klebsiella, and Salmonella. So these bugs are responsible for a large number of antibiotic resistance deaths, and indeed, they're very, very resistant bugs, and getting more so. So we're already having trouble treating disease caused by these organisms. So what did we do? Well, we thought, let's look at the bacteria that we carry. Can we find new ways to prevent or treat infection using those? So what's the experiment? As I say, it's very simple. We took 100 of the different types of bacteria that live inside the human gut, and we just grew them one by one with the pathogen. So we're just going to put the pathogen in, we put the other strain in, and we see how well does the pathogen grow. So that's the key experiment. The one thing we did in addition to this, where we thought, well, maybe they work better in teams. So we put together groups and combinations of different species of bacteria and then put them in with the pathogen. Okay, so that's the basis of the experiment. So what do we find? So I'm just going to show you one graph to give you the key result. So along the bottom here, what you're seeing is an increase in the number of species we're putting together in one of these mixtures before we add the pathogen. So 0 means it's just a pathogen growing on its own, and then 50 means we put 50 different species of human gut bacteria into the experiment and asked how the pathogen grows there. And on the side, I'm going to show you how well the pathogen grows, so how much pathogen growth you get as a function of all this, is this slowly increasing the diversity. So what do you see? Well, you see quite a clear result. The more different bacteria you put into a mixture, the less well the pathogen grows. So what is this teaching us? It's teaching us through an experiment that having a diverse microbiome is a good thing. It helps you protect you against pathogens growing inside your gut and in other parts of your microbiome. But of course, the key question is, why is this? Why is this going on? So we did quite a lot more work to work out what was going on, and actually at the heart of this is a very, very simple explanation. And it's all about competing for nutrients inside the gut. So I'm just going to run through what's happening, because it's, as I say, it's simple, and it also gives us the potential to predict which bugs are going to be the best against pathogens. So, in our cartoon, imagine we have a pathogen, and it can eat 4 different things. In practice, they can eat many different nutrients, the pathogens. It's not just 4, but let's assume it's just 4. And while the nutrients are simple things like a sugar or a fat, we can think about them as, say, 4 different foods for the pathogen. So in this example, imagine you only have 1 strain of bacteria living in your gut. The chances are it won't eat all the different things that the pathogen can't eat. As a result, the pathogen grows quite happily. It has the food it needs, it grows well, you don't because you get the disease. Now imagine you have a diverse microbiome. Now, under these conditions, the bacteria that are already present in your gut have consumed all the different types of nutrients that the pathogen needed to grow. Now, the pathogen's got nowhere to go, nowhere to grow, and as a result, it's unable to cause disease. So our experiments showed that this effect is extremely powerful and extremely effective in preventing pathogen growth. And so we called it nutrient blocking. The idea being that having a diverse microbiome allows you, most of the time, to block the entry of pathogens simply because those bugs are preventing the pathogen from growing. So we recently published this, and we're excited about this, not just because it helps us understand what's going on inside the gut, but also because it gives us the ability to predict which people's microbiomes might have gaps in them that need to be filled with beneficial bacteria in order to limit the probability of pathogens growing. So it's helping us move from a descriptive science to a predictive science, and that's where we want to be with the human microbiome. Okay, so that's about potentially preventing infections, but that's not enough. When we take antibiotics, we're typically treating infections. So you already have a standing infection. Can we also use bacteria there? And I would argue again, I think we can. So what about curing disease? Can we do it? I think so, but we still have work to do. Now, in order to do this, I have to take you back to those first particularly aggressive examples of bacteria fighting each other. Thinking about nutrients will probably not be enough to cure disease. If a pathogen is already in your system, it's found something to grow, it's causing problems, and it's too late probably just to think about competition over different nutrients. However, we can take a bacteria that makes a toxin, its own antibiotic, that will target the pathogen. And importantly, many of the antibiotics that are made by bacteria are very, very specific. So if you take an antibiotic in the hospital or from the doctor, they tend to kill everything. They're very broad. They affect a lot of different bacteria. But the antibiotics made by most bacteria are very targeted and specific. So we can identify strains of bacteria that will make a very specific antibiotic that will target a pathogen without upsetting all the other bacteria that potentially can actually help limit the pathogen and cure disease. So that's what we're doing. We're identifying those types of bugs and those types of antibiotics, and we're starting to do our experiments again. So this work is still early days, but I'm just going to give you one example from a test tube where we took one of these pathogens, a dangerous antibiotic-resistant bacterium that's hard to treat using normal antibiotics. and we added to a culture one of these good bacteria that is making its own toxin that targets the pathogen. So in this, another simple experiment, we start the pathogen at very, very high levels, and we start our good bacteria at very, very low levels. But now watch what happens over time. Because we had to think about nutrients and make sure that the good bacteria can grow, and indeed it can under these conditions, it can still compete for nutrients. So it comes up in frequency, and as you'll notice, once it gets high frequency, it's then releasing these toxins, and that's knocking the pathogen right down. So this is an example then where we can introduce a bug that's harmless to us, doesn't affect many of the bacteria in the gut, but will very strongly inhibit a pathogen. Okay. So in summary then, bacteria do cause diseases, and this obviously is a huge problem and something that we have to find new ways to treat given the rise of antibiotic resistance. What I've argued today is we also need to recognize that many bacteria are good for us, and it's these bacteria that we should be focusing on as an alternative to antibiotics. Thank you.